Thank you, Jenny. It's my pleasure to welcome Monique. Monique is currently working at the Monterey Bay Aquarium Research Institute, and she has been since 2014. She got her PhD in 2006 from the University of Toulouse in physical and biological oceanography, and her research focus on ecosystem processes and the interaction between the physical and the biology in the coastal and global ocean. And today she's going to be talking about ecosystem hotspot in the California current system and the physical driver. And she recently joined the post panel. Thank you, Monique.
Thanks, Cecile, for the introduction. I'm pretty happy to talk to you today to present some of the work I've been recently doing that I think is of most interest to the panel. And so what I want to talk about, let's see. Oh, I need to be able to, okay. Those are the type of hotspots I'm going to talk about today. So those are places I'm looking at the California West Coast where there's a lot of biology. So you can have whales, you can have seabirds, you can have sea lions, you can have people on boats who come to watch them. And so there's a lot of implication for both economy and fisheries and conservation in general. And so we need to understand where, when, and why those form. This image is a little bit misleading in the sense that this is a very small scale hotspot, and I'm talking more about mesoscale size hotspots, so those large areas that are more biologically productive. And I will be talking about a couple of little studies that also highlights different mechanisms by which the physics control the biology. First, I'll talk about krill hotspots all along the California coast. And then I will be looking at time series in Monterey Bay and around to look a little bit more at temporal variability. So the hotspots, when I talk about krill hotspots for starters, this is how we can observe them. This is a result from a yearly survey that NOAA does, usually in May, around May and June. And this is based on acoustic data underway. And as you can see, there's different locations where you have more hotspots occurrence. And those are the three main hotspots that I'll be referring to today. That's there's one off of Cape Mendocino, one near San Francisco, Monterey Bay, and one off of Big Sur. And the other thing we know about those is that they are coinciding with hotspots of top predators, which is not too surprising because they're an important food source for all those predators. And this is a picture that is based on tag data that shows where those predators aggregate. So that's very well, and we have fairly good understanding of where those hotspots are. But when you start to look a little bit more about the data at any given time, it's not an easy picture. On the right is an example survey back in 2006. And so that's the acoustic data I was just telling you about. And as you can see, it's extremely patchy, and you have very high concentration next to very low ones. It's a little bit difficult to get a good picture of it, plus it's only once a year. And on the left is a screenshot I took from the top website, which is the program that deployed those tags. And every yellow dot is a tag, so there's just not a lot of them at a given time. So we don't really have the data to be able to track those hotspots. And that's where we need to understand why they form so that we can predict them and understand how they change. So the first method we used to look at that is a coupled Ramsey-Nimuro model. This is a study that just got published in JRL. And so Jerome Fichter at UCSC is the one who is doing that work. So this is a model that's being forced on the outside by a 1/10 of a degree data assimilated model. And we're looking at a smaller domain, which is the one here on the left, that temperature output, and it's 3 kilometer resolution. And on the right, you have the result for chlorophyll, and all of the black dots are the locations where we have net trolls from that same NOAA survey. So the model does a very good job at representing the krill hotspots. Here you have on the left the result from the model, and here you have the observation. So those 3 hotspots I was talking about before are being reproduced correctly. And when you look at temperature variability against those yearly surveys, in this case against troll data, you also have a pretty good correspondence. So basically, we are able to represent the model, we're able to represent the data even though they're pretty sparse, and the model gives us much better spatial and temporal coverage. So we learned a few things thanks to RUMS. One is that the location of the hotspots is strongly driven by surface currents. Here you have latitudinal profiles of krill and chlorophyll, and as you can see, they're offset in May and June, which is because those currents are strongly southward, and so they displace the water mass south of the high chlorophyll regions. Whereas in July and August, when currents are weaker, they become collocated. And the other thing is that winds, I didn't put the figure here, you can look at the paper if you're interested, but winds are what drive the interannual variability of those hotspots. And the second thing we learned is that now that we have that better view of what the hotspots are like, they really match with predators. And so on the bottom left, you have a whale species, and on the bottom right, a seabird species. And that's something we had seen from observations, but that's kind of being confirmed by that, so that those krill hotspots really are ecosystem hotspots. So the model is extremely useful because we have that very good representation of hotspots. The main issue is that it requires a coupled physical biological model. It requires being forced by a data assimilated model, so it's pretty heavy. And so we wanted to find a way to kind of simplify that and see if we could represent those hotspots using a lot more simple relationships and maybe using just satellite variables. And this is what I call the gross advection model. And the idea is that you take a water mass that starts at the coast that's being fertilized by upwelling, and you track it to see where it goes while you look at how biology develops within it. And so it's kind of the same concept. It is the same concept that was introduced by Botsford et al. Back in 2003. It's the conveyor belt is how he called it, I believe. The difference is that he was just looking at the water mass going offshore, whereas we're following the real currents. And to show you a little bit more how that works, you have here a representation of the plankton model. So you start at time 0 right here on the left by nitrate supply, which is something that you can calculate by just multiplying upwelling by nitrate at depths. And you have only that instant, that beginning nitrate supply at the beginning, and then you just let biology run. And so you have first a peak of diatoms as they consume that nitrate, and then zooplankton starts to grow later. So I have 2 phytoplankton, 2 zooplankton, but the one that interests me is the big zooplankton here, the black line. And as you can see in this model, it peaks about 3 weeks later. So when you have that little model, you can plug it to currents. And this is that image on the left, and I'll show a movie of it on the right in a second. So we run this at 3 kilometers along the coast, following the RUMS resolution. This is being forced by the RUMS model outputs, meaning the winds that force the model and the surface currents from the model, or top 30 meters rather. And the reason is that we wanted to see if that method can reproduce the Nemiro hotspots. So on the right, you have that little movie where the bottom line represents the temporal evolution of krill. And then I have trajectories that start at the coast. And then that krill that develops in all of the trajectories as they are being advected by the currents. And the concentration in each trajectory is a function of the nitrate supply at the beginning, which is why they're all different. And the last step is that you can then combine all those runs. So those are, sorry, I want to advance. There we go. Those are runs, as I said, they're initialized daily at every 3 kilometers along the coast. And so if you want to look, for instance, on May 2009, you can look at all of the runs that were initialized starting in early March until late May. They last for 2 months. And pick the data points that match that time frame and put them on the grid, and that's how you get those maps. And despite being a very simple method relative to a full assimilated RUMS Nemiro model, It actually works pretty well. Those are comparisons where on the left you have results from the Nemiro krill, and on the right you have that gross advection method. And so as you can see, the main hotspots are reproduced in the correct locations. And the timing, we have a seasonal propagation, propagation of the hotspots that is being reproduced as well, with the bottom, the color is the months of the year when they peak. And we also are able to reproduce their interannual variability. Those are time series in the 3 hotspots, and that are being taken at the peak months for each of them. And so, as you can see, there's actually a really good agreement between them. So it's not too surprising because, as I was telling you earlier, winds and currents are really what drive those hotspots in the Nemiro model. But it's kind of nice that just being so much simpler, you can still reproduce it. And since it is simple, it also means you can get it from satellite, because you can calculate upwelling from satellite winds or reanalysis. In this case, I'm using CCMP. And surface currents, I'm using the globe current product. And so you run exactly the same thing, and those are the time series I get in green. It's not exactly the same as Nemiro, which makes sense because the forcing is different. So we're in the process of validating against data, and this is a comparison for the central hotspots, where you can see that there's a pretty good agreement between the prediction from that model and the krill in May. So that was my first story, and the second one is really about other species, because as I told you, those hotspots coincide with stop predator hotspots, but it's a little bit more complicated to know about their temporal variability. And so to look at that, we've looked at 3 time series that MBARI has been collecting for a long time. And there's been a recent publication in Oceanography for MBARI's 30 years that gives a lot of detail about this time series and other work on MBARI if you're interested. Just briefly, we have 2 time series that are in Monterey Bay for the surface and midwater animals, and one that is off of Pond Conception, all the way here, that's the White Star, for the benthos. And those are the type of animals I'm looking at. So for surface, it's phytoplankton. For water, it's mostly medusae, and there's some fish, and it's from video, so whatever you can see between 200 and 1,000 meters. And for the benthos, it's mostly echinoderms, the species that are under the seafloor. So just to show you what they look like, the surface and benthos started in 1989, the midwater in 1997. For the surface, I'm looking at just water samples collected at the surface and analyzed by epifluorescence microscopy and flow cytometry. And we look at the species, their sizes, estimate their volume, use some carbon to volume conversion to get at plankton biomass per phytoplankton groups. The midwater uses ROV video transect. So the ROV stays at a given depth for 10 minutes, and everything is being imaged with the camera not moving, so you can estimate the volume that's being imaged like that. And then somebody in a lab identifies every single species that's being observed, and you can estimate animal density. And I'm looking at an average of transects between 200 to 1,000 meters. And they are every 100 meters, but I'm only looking at that vertical average. And finally, for the benthos, it's also from images. First, it was towed camera sleds, and starting in 2007, it's ROV video. And the main difference between this one and the others is that its temporal resolution is not as good. The ship goes out every 1 to 2 years, whereas for the surface and midwater, it's every 3 to 4 weeks. And so to analyze those time series, we've looked at principal component analysis. So we just removed the mean and applied a transformation to avoid putting too much weight on high values, but the seasonal cycle is still in there. So you probably all know what principal component analysis does, but in which, just in case it pulls the time series that explains the most of the variability, the most of the variance for the time series that goes in it. So here's an example for the benthos. I only have 4 species. Those colors are the group they belong to. So the top panel is the time series. and that principal component for the first mode. And the bottom panel is the loadings for each species. And so the way to read it is that the strongest the loading, the more they are correlated with that time series. And also the sign of the loading tells you if you're positively or negatively correlated. So in this case, because all of the loadings are of the same sign or very weak, that really means that that principal component captures the entire community or most of it responding synchronously in the same directions, which means that when the time series is positive, you have a lot of animals, and when it's negatives, you have a lot less animals. This is what we get for the midwater. There's a lot more groups here. There's about 90 of them. And interestingly, you get the same type of results with most of the loadings being of the same sign. So again, that time series here represents a lot of animals when it's positive and a lot less animals when it's negative. And finally, for the surface, same story again. You have some negative loadings for picoplankton, which is the type of species you find in low productivity situation. So that principal component here is actually pretty strongly correlated to total biomass and other independent productivity estimates like primary production and chlorophyll. So that for the 3 time series, my first mode captures changes in biomass or density, like the productivity of the system, if you want. And so what we wanted to do was to relate that to upwelling, because we're in an upwelling system, and that's what is supposed to be driving the productivity of the system. And it's also the one driver that we found to work the best. The issue is that, as you can see, there's a very big difference in terms of the variability. Upwelling is a daily time series at the wind station that is noted by the black dots here off of Monterey Bay. I will use the one in Point Conception for the benthos, and it's super high variability, very high frequency, whereas the surface is more seasonal, and then midwater and benthos are more interannual. And the way to relate that comes from a paper back in 2013 by Di Lorenzo and Oman, where they talked about integration of a forcing by the ocean and by biology. And so a way to look at it is that when you have a very high frequency forcing, such as what you get from the atmosphere, it takes some time for the ocean and then the biology to respond to it. And that's what's being called the integration of the system. So there's some sort of a damping time scale that's inherent to what you're studying that kind of slows that response. And you can calculate it using that simple equation that you have here on the left. So in my case, I'm not looking at the double integration like they did in this paper. I'm simply looking at integration of upwelling and comparing it straight to biology. And so I found the timescales that work the best. And it's about 2 weeks for the surface, 2.5 years for midwater, and a little bit over 3 years for the benthos. And this is what you get when you take that upwelling time series and you integrate it by those corresponding time scales. And so the longer the time scales, the more the initial signal is being damped. And so the more you have low frequency that just appears. And so this is what happens when you compare it to the principal component. You actually have a pretty good correspondence. And so that shows that upwelling is able to control biology by that integration, which is not something we had for the krill, because krill is a lot lower in the food chain. And so, as I said earlier, in my model, it picks 3 weeks after upwelling. So it's kind of similar to, it's a little bit delayed relative to phytoplankton, but it's still a very fast response. But when you start looking at higher predators, it's high level predators, that's where you really need to take into account that integration. Okay, so just to conclude, I wanted to highlight 2 methods by which you can match physics and biology. One is to consider advection and how the populations evolve over both time and space, which is very important in a place like the California current where the circulation is so strong. And the other one is the integration, how biology takes some time to respond to the physical forcing, and you need to take that into account too. So in both cases, upwelling is a very strong driver. And I didn't talk about surface currents for the time series. It's a little bit more complicated to assess because we just have point measurements. But yeah, and what else did I want to say? I guess the main point about all of that is that once you start to identify those drivers, you can start to understand how climate might impact those biology. And so you can start to see what would happen in the future. And actually, we're starting to do a study where we would look at projections of winds, currents, and try to see what the hotspots would look like in the future. The issue is that those, both for the krill hotspots and for the time series, they're very sensitive to the forcing. And so it's not clear at this point whether you can predict winds and currents with enough accuracy to be able to really detect what those changes would be. And just to conclude, I want to acknowledge both the funding and my co-authors. So most of that was funded by MBARI and NASA for the zooplankton hotspot work. And so this was done in collaboration with collaborators at UCSC and NOAA and MBARI. So Jerome Fischer is at UCSC, Jared at NOAA, and all the other ones are at MBARI. And that's all I have. So I'm happy to take questions.
Thank you, Monique. Reminder, if you do have a question, use the raise your hand button. Otherwise, you can type your question into the chat box and I'll ask it out loud for you. All right, see, Anish has a question. Go ahead, Anish.
Can you hear me okay?
Not very well.
Oh. Yeah, you're a little quiet.
Okay, let me try this. How about now?
It's better.
Okay, great. Yeah, thanks a lot. It was really interesting. So I was wondering about the comment you made in your concluding slide, also related to the ROM's Nemuro work in terms of how climate variables and climate variability can actually help improve prediction of these hotspots in the ecosystem variables, right? And in your work, you look at upwelling locally in those regions. I was wondering, have you also looked at like boundary forcing into ROMs if you have like a El Nino year versus a La Nina year, or if you're in different phases of like the large scale circulation in the Pacific, how does that influence the prediction skill in the model?
No, it's not something we've looked at. I think, I don't know if it answers your question, otherwise let me know. What I think is like, if you look at, let me go back to this one. So the reason why I was saying that, like that time series of Upwelling is very high variability, right? But when you start to look at integration, it's really that low frequency that shows up. So it's not clear to me, and it's not a test that I've done, whether that low frequency variability is enough to be able to predict what the biology is doing. One thing I've noticed is that if I try to relate the benthos to the wind station of Monterey Bay, even though they're not that far apart, it does give a different answer. What I have not tried to do, and something I need to look into a little bit more, is pull out the low variability, the low frequency in the Upwelling time series and see how it compares to those integration. And in other words, I'm not sure how much the very high frequency drives the biology response when you start to have long integration, or if you really just care about that low frequency, in which case, being able to predict things like El Nino and large scale climate is sufficient. Does that answer your question?
Yeah, it does. And yeah, that would be really interesting work, I think, to look into that. Thanks.
All right, so we have a question from Shane.
Hello, Monique. Can you hear me?
Yes.
Thank you very much for taking the time to show us your work today. That was very interesting. I have a question. At the beginning, you showed a map maybe that shows the location of all the canyons. along the coast. And then I think one of your conclusions from the growth advection model compared to ROMs was that you were doing a very good job by predicting upwelling based on winds only. So would you say that the details of the topography doesn't matter very much?
No, I think it's a matter of scale. And that's one thing that we've been wondering about. So a first version of that work was published in Journal in 2017, and that was something where I just used the seasonal currents, so it was a lot more simple. And we also noticed, like, we were still able to reproduce the locations, and we wondered to what extent the canyons have an impact, the canyons and the topography in general impact the currents, such that you really have both, like like it could be just a coincidence in a way that the currents bring the zooplankton in places where there are canyons, because it's just the way the topography influences the currents. And the other thing is simply that scale. It's like you're looking at different scales if you look at a canyon versus if you look at something that's mesoscales that's about 100 kilometers across also.
Okay, so it depends on the scale you're looking at, basically.
Yes, so it depends on the scale, and even when you look at the mesoscale, it may be that the canyons drive the circulation in a way that it still congregates the hotspots in those locations as well. But it's something, I don't know, like Nemiro, even at 3 kilometers, I don't think RUMS represents the canyons well enough to really capture that effect. So, yeah.
Okay, great. Thank you very much.
All right, we have a question from Allison.
Hi, Monique. Can you hear me okay?
Yes.
Yeah, great. Thanks for your talk. Yeah, I was just wondering if you have thought about how these factors might apply in other upwelling regions in the globe.
Yeah, so that study I was talking about the seasonal cycle, I actually did it in the 4 upwelling systems. The main issue you run into is that there's just not a lot of data. And so when you start to look at places like the Canaries in particular, there might be some data, but either they're not public or they're not published, or it's really hard to get them. So as part of that work, I had tried to look through the literature to find where those hotspots were, described, and it was pretty challenging to do. So there's a few that I found that was kind of supporting the type of results I was getting, but not many. And when it comes to the time series, I believe it's pretty unique. I'm not aware of any other time series that monitor, especially the midwater and the bentos, for such a long time and so consistently. So it's I just don't think we have the data to really answer that question, because you can apply the method, but if you don't validate it, it's a little bit more challenging.
Okay, thanks. Yeah, the data set is really, really unique, I imagine.
Yeah.
And I think we'll conclude with one final question from Mike. Go ahead, Mike.
Hi, Monique. Yeah, this is really interesting presentation of the biophysical interactions. Can you speak to the biogeochemistry in the system and its importance, and how that's related to upwelling and the other physical properties that can in part drive the response here?
Yeah, so for the 3 time series, not really. I actually did some tests trying to look at nitrate supply as a forcing instead of upwelling. So combining upwelling with nitrate at depths, I did not succeed. It didn't work as well, and I'm not sure why. It could be that we don't have the correct data, and it could be that it's just too precise in a way to be able to tie those together. For the hotspots, it's something that we've been looking at, because the nitrate supply I used to force the model is upwelling multiplied by nitrate at depth. So that's where that biogeochemistry is important. And that's actually one thing that's very useful for the gross advection model, is that you can really decouple the different forcing. It's not something you can do with, well, you might be able to do it for nitrate, but for instance, winds versus currents, the winds are driving the currents. You cannot separate their importance. Whereas for the gross advection, I can hold one steady or rather climatological and let the other one vary interannually. And so when I do that for nitrate versus upwelling, upwelling is really the strongest driver. You get almost as good as a result if you hold the nitrate supply seasonal and keep the winds varying. And if you do it in reverse, that just doesn't work. So it's something that I need to spend a little bit more time, like, to be able to quantify that a bit better. But the first impression is that upwelling is stronger of a driver than nitrate at depths.
Thanks.
All right, so I said that we would conclude with one more question, but do you have time to take one more? I see Antonietta has a question.
Sure.
Thank you, Jenny, and thank you, Monique. This was really fascinating, especially the time series of the species and try to relate them. So, as you said, upwelling is such an important quantity in this area, but have you also looked at other physical quantities, like maybe just temperature, which probably is somewhat related with upwelling, but I don't know, may provide slightly different information? So I think is the upwelling important just because of the nutrient supply, or also just because of the environment affects the other physical environmental conditions?
Yeah, that's an excellent question, and that's one that's still being debated, which is the reason why I didn't get into that too much here. That's something that we're discussing with my co-authors. So I did look at other predictors. We have a lot of data that are being measured in Monterey Bay, and I also looked at global scale forcing, like the NPGO and PDO and El Nino and all that. And upwelling consistently worked better, and better than something like local temperature, for instance. This being said, there's clearly a link with some large-scale forcing. A big one is the NPGO that actually match pretty well with the midwater once you remove the seasonal components and the trend in particular. And so at this point, it's not entirely clear if it's a direct connection between upwelling and the ecosystem, or if there is something in the environment that's tied to upwelling that could be temperature or circulation or something else that is really what drives it. And so, yeah, it's something that we're still trying to assess. It's not an easy question. I don't know if we can conclusively answer it.
Yeah, it's a hard question, but it's very fascinating. Thank you.
Thank you.
All right, great. I think that should conclude our pause panel webinar today. So thank you everyone for joining us, and thank you to Monique for giving the talk today. Our next pause panel webinar is scheduled for September 3rd, and it'll feature another new panel member, which is Jingfeng Lang. So stay tuned for more information on that.